Hello, everyone. My name is Jade Cronin. I'm a PhD candidate over here at Clemson University, working in the Children's Lab. And today I'm here to give you a quick little talk about some of my dissertation research, primarily some preliminary research that we are about to publish that some of my creative inquiry students have actually helped me with. So the title of this presentation is Crayfish and Their Hidden Tenets: Thermal and Symbiotic Impacts on Crayfish Behavior. Oops. Now, as we all are probably well aware, the world is rapidly changing, primarily due to the warming of the climate, as well as different anthropogenic or human effects that we have on our different ecosystems. Now, these impacts may include things like reductions in species survival, loss of habitat suitability, which is actually demonstrated in our figure over on the right. So if you look on this left image right here, what you'll see is that there's actually multiple different colors, with purple relating to loss, this goldish color relating to stable, green indicating a gain of territory, and blue indicating a vulnerable territory. Now, under just climate change, we will see that there's a very sparse amount of loss, but there is a lot of different vulnerable habitats that may be lost for this organism, greatly shrinking the available area it has to live. Add in anthropogenic changes such as human population density, and we see that the loss of habitat is significantly increased. On top of this loss of habitats and species survival, things like the community composition, species distribution, and our overall biodiversity may be greatly altered by anthropogenic changes and climate change. There's one other area that is often understudied and not very much thought about, but that is interspecific interaction. So human factors as well as warming climate may actually alter these interactions. And you may be asking, what do you mean by interspecific interactions? When I refer to interspecific interactions, there are three primary interactions that I am referring to. The very first one, which you can see in our image over here, labeled 1, is predation. So predation is typically when a predator or an organism kills and eats another species to actually feed, get nutrients, and survive. Followed by that, the other interaction that's very common is competition. So competition is typically some sort of interaction between two species on a very similar trophic level where they actually compete for a limited resource. So this limited resource may be something like habitat space or territory, dens, or even food, which can be demonstrated in our figure on the left, where you see a squirrel and another organism fighting over a nut. Now, for the purpose of my talk, I'm going to be focusing on the third type of interaction, which has received relatively less focus in terms of a climatic context, and that is symbiotic relationships or symbiosis. Now, for the purpose of the study, symbiosis refers to close and long-term biological interactions between different organisms. So in the figure on the left, we actually see a rhino and a bird, and this represents a symbiotic relationship in the fact that the rhino actually allows the bird to clean it or feed on it um, throughout the time so it does not have to clean itself, and the bird actually gains a benefit from that by having food and somewhat of a protection by being on that rhino. Now, symbiotic relationships have typically been relatively misunderstood, despite the fact that they typically have a very large impact on the community and ecosystem assembly of an area. Historically, we've relatively misunderstood these relationships and thought they were relatively static in the sense that they never change in terms of their fitness benefits. So if you look over on the left for figure A, we see we have species 2 on the x-axis and species 1 on the y-axis. And you also notice we have a plus sign, minus sign, and a zero sign, with the plus sign correlating with a positive fitness benefit, minus sign a negative fitness benefit, and the zero being no change in fitness at all. So these relationships include things like mutualism, where both organisms have a positive effect on their fitness, parasitism, where one organism has a negative impact and the other one has a positive impact, and commensalism, where one does not have a impact at all. So there's no negative or positive for this interaction, but one of them greatly benefits from it. Now, we used to think that these were static, but now we do realize that they rely on a dynamic continuum, which you can actually see over in figure B, where these relationships can actually shift anywhere from parasitism to mutualism to commensalism based on the organisms involved and the environmental context that the relationship is occurring at. So, my project focuses on crayfish and brachiopodelid symbiosis, and there's a number of reasons why I've chose this system. The first being that it has been proposed to be the ideal model system for freshwater symbiotic relationship research, primarily due to the fact of the ease of husbandry and ease with working with crayfish both in the field and the lab. But on top of that, these crayfish actually demonstrate a unique regulatory behavior of the symbiotes through grooming, which is something we're going to explore a little bit more. On top of that, crayfish are actually very ideal for examining the impacts of anthropogenic change, primarily because they're freshwater organisms. So, freshwater systems are typically those that are under the highest pressure from anthropogenic change due to humans being typically very close to those. So, these crayfish are constantly dealing with these change and allows us to actually look at the impacts humans and climate change will have on them. Finally, crayfish typically have multiple symbionts on them at a time, so this system actually allows us to examine multiple symbiotes, primarily brachiopodellids and ostracods, whereas other systems typically are not looking at multiple different symbionts. Now, crayfish overall are just generally a very important and um, ecological organism, but they're also very important for our economy. So these organisms are typically keystone consumers. They're generalists, where they actually eat pretty much whatever they have available, typically feeding on detritus and algae, but they're known to also eat macroinverts. And if stuck or have the opportunity, they will also eat things as big as snakes. On top of that, these are known as ecosystem engineers. So crayfish typically burrow and do different activities that actually change the ecosystem that they live in and provide different things like habitat or dens for other organisms that would not be there if they were not present in the ecosystem. On top of that, crayfish are actually very threatened by multiple different anthropogenic factors. So they are a very important organism for us to look for, especially given the fact that we have multiple fisheries that rely on crayfish to function. So, for the purpose of my study, I'm going to be looking at a number of different organisms. So if you look at the top left, you'll see we have an image of a crayfish, and this is our first focal organism, which is Cambarus latymanus. So Cambarus latymanus, or the variable crayfish, is a crayfish that is typically found throughout South Carolina and the Southeast. It is a very abundant organism, and it can be considered a habitat specialist. Our second organism would be that of Cambarus bartoni. It's another organism that is found very much throughout the South. It's extremely common in all of our streams, but this is actually more of a habitat generalist, with their range spanning all the way from Canada down to Florida. Now, for the purpose of this study, we're also going to be looking at multiple different symbionts. The first one, if you look at the bottom left, we have our ostracods. So ostracods are known as sea shrimp. And for freshwater ostracods, we actually don't really know much about them. The most we know is that they are actually commensals of some type or symbionts of some type with crayfish, but we do not even know if they're obligate symbionts. So there's a wide area of research that needs to be done on these organisms. The second symbiont, which is a little bit more researched and more common when referred to crayfish, is that of brachiopodellids. So these are annelid worms that typically function as cleaners for these crayfish. There's been numerous studies that have actually shown that they defile the gills and clean their carapaces. That actually allows the crayfish to grow at a much higher rate than when these organisms are absent. However, there are also studies showing that these organisms do not always provide that benefit. So if you look at our image right here, you'll see that some of these brachiopodellids are actually in the gills, which I will highlight right here, as well as laying eggs in these gills. So at very high densities or under very low fouling pressures, we actually see a shift between the benefits from the brachiopods to actually some negative impacts on the crayfish. And that is primarily through, if you look at the bottom right, gill scarring. So when they are in these gills, these brachiopods actually will start consuming the gill material. So if you notice, you'll see that we have some very brown areas on these gills. And these are actually melanization scars that occur when these brachiopods start actually consuming the gills because there's not enough detritus and other things for them to consume. Now, for this presentation, we have a number of different objectives that we are going to be looking at. This is going to be very, very brief compared to the scope of the actual work that we did. But the first objective is we wanted to look at the relationship between body size and symbiont density, where we actually predicted that a larger body size would positively be related to symbiont density as there's actually more microhabitat space. And there's some literature actually demonstrating already that this is the case. After that, we actually wanted to look at the relationship between gill scarring and symbiont density to ensure that there's actually some relationship between those and the gill scarring is not caused by something else. So we predicted that gill scarring and symbiont density will be positively related to one another. Now, moving into our behavior, we wanted to look at a couple things. So first, we want to look at the thermal impacts on behavior. We assumed or predicted that increasing temperature would actually increase the frequency and duration of our grooming behavior. And this was primarily due to the fact that there's a large amount of literature showing that increased activity and behaviors in macro inverts while temperature is increasing. Following that, we wanted to look at if these symbionts actually had an effect on crayfish behavior. And we want to look at that in two different ways. The first way was just looking at the relationship between symbiont density at the time we looked at them and crayfish behavior, where we predicted that there would be a positive relationship between symbiont density and grooming frequency and duration, specifically because this is a behavior they actually use to regulate these symbionts. Because symbiont density is relatively a snapshot only, because we only have the symbionts that are available at the time, we also wanted to look at the relationship between gill scarring, which is somewhat of a proxy for overall symbiont activity or symbiont damage, with crayfish behavior, where we also predicted that gill scarring would correlate positively with grooming behavior and grooming duration. So, what we did was we first obtained distribution data of both species of these crayfish from the Department of Natural Resources here in South Carolina and the Global Bio or GBIF, which is the Global Biodiversity Index, which had a numerous amounts of distribution data, and we plugged those into ArcGIS. So we specifically wanted to look at the Clemson experimental forest. So what I did was generate random points of occurrence where both these species were found and selected three different streams from those randomly to actually sample from. And from that, we selected Wildcat Creek, which can be seen right here in figure C, Turkey Creek, which can be seen in figure D, and Todd's Creek, which can be seen in the B portion of this figure. Oops. So what we did was we actually headed out to those different field sites and we laid a number of different traps so we could actually catch these crayfish. Now, our experimental design spanned over 120 meters of these streams, where we placed one trap every 30 meters, having 5 different traps at each of these streams. We baited these traps with cat food to ensure that we would actually get crayfish into those. And every week, me and my students, which you can see one right here, this is William. He's my field expert who comes out with me frequently. But we went, collected these crayfish, and we actually measured sex and identified them using the guide provided by Ebersole and Jones 2004. Once we did this, the organisms of interest, which was our Cambarus bartoni and Cambarus slidemanus, we brought back to Clemson University. From there, we ran a number of different behavioral trials with those. So we started off with a relatively short range of temperatures, focusing on 5 different temperatures ranging from 68 degrees Fahrenheit to 76 degrees Fahrenheit in 2-degree increments. And what we did was we set up a number of tanks at these temperatures, And we would place these crayfish in for 2 hours. One of those hours would allow them to acclimate to the temperature so we didn't have any shock influencing their behavior or change in their environment influencing their behavior. And the other hour, we used a CCTV camera to actually film those trials and see how those crayfish were behaving. Once we've actually got those videos, we used Boris, which is a free behavior tracking software on videos to look at a number of different behaviors, primarily locomotion, grooming, posture, feeding, and other, which was typically construction behavior. But for the purpose of this study, I'm primarily just going to be focusing on our grooming results. Once they were done with the trials, we had to actually euthanize those. So if you look over at the image on the left, you'll actually see kind of one of our euthanation methods. So what we would do is we would create a giant ice bath for these crayfish and put them in there. Now we would wait till they actually ceased movement and stopped moving, or if they've hit a point of physiological death where they could no longer function, and left them in there for a period of time before determining that they were dead. We then placed them in ethanol in 50 milliliter conical tubes so we could store them and actually dissect and look at the symbionts in gills later on. So once we had them in these tubes, we actually moved them over to a stereodissecting microscope, where we actually conducted a series of thorough dissections of these crayfish, specifically looking to count the number of symbionts that we found on those. While we did count all symbionts we found on them, for this, I'm primarily just going to be focused on our brachiopodellids and ostracods that we see on them. We did find numerous other ones or other diseases, such as burn spot disease, which you can see at the bottom left, which is these burn marks that you can kind of look at the tail. But we did not account those into any of our analyses that I will be presenting today. Following that, we actually removed the gills from all of these crayfish and stored them in a 15 milliliter tube with ethanol before we were ready to actually photograph them. So when time was to actually analyze these, we took each individual gill filament and used a digital electron microscope to actually take different pictures of those organisms, looking for things like brachiopodellids and eggs, as well as gill scarring on those. Once we had a photograph of both sides of each gill filament, what we did was we uploaded those images into ImageJ, where we would actually look at the average gray values found in the entire image to actually determine the level of scarring that was occurring in those. And this was following the method of Rantal and Quartet from 2004. They had used this as an immune reaction test for crickets. Now, once we completed all that, we had some simple amount of statistical things that we needed to do, but the very first thing was to ensure that we had a sufficient sample size for both of our species. So I ran a power analysis, which indicated that we do in fact have a sufficient sample size for both species with a number of replicates of being only 8. However, we then ran an ANCOVA to see if we could actually just combine those species or if species had actually any impact on our results. And we actually found that nothing, there was no species effect at all. So for the purpose of all of these results, we actually combined both Cambarus bartoni and Cambarus latimanus. We then just used JUMP Pro 18 through the school to actually analyze all of this. So our very first thing we wanted to look at was whether there was a relationship between body size and symbiont count. Previous literature had actually indicated this, and based on our predictions, we thought that there should be a relationship that we see. But if you look over on the graph on the right, you'll see on the x-axis we have the square root total symbiont, and on the y-axis that we have our carapace length, which is the standard measure used for crayfish. And you can see that we actually found no significant relationship at all for this. So there was no relationship for body size and symbiont count. After this, we wanted to look at, well, was there a relationship between symbionts and gill scarring? We had predicted that yes, there would be, given the fact that these symbionts typically are associated with causing the gill scarring. So for this analysis, you'll see we have on the x-axis our gill score, and on the y-axis we have 3 different variables actually. So we have in blue, we have the square root of our ostacod density, we have in our red, square root of brachiopodella density, and then green is our square root of total symbionts. And what we actually found by running this simple regression is that Total symbionts, brachiopods, and ostracods were all positively correlated with gill scars. Following that, we ran a simple ANCOVA just to see the impacts of temperature on symbiont density, or we ran a simple ANCOVA where we looked at our independent variables being temperature and symbionts, with our dependent being gill score, and we used species as a covariate to see if that mattered at all. And what we found was actually that the only variable that was related to gill scoring was actually our total symbiont counts. Following that, we moved into our behavioral results, and the very first thing we wanted to look at was the influence of temperature on behavior. Now, for this, we ran both frequency and duration, or occurrence instead of frequency. Those are both the same term. And we have our temperature values on the x-axis, and we have occurrence and duration on our y-axis. And from this, we can see that we actually do not have a significant relationship. However, we do see that there is a negative trend for these with duration approaching significance specifically, suggesting that maybe there is something there and it's something that we need to look at in the future. Following that, we wanted to actually look at the relationship between behaviors and actual gill scarring, which is that long-term symbiont effect. And what we actually found was there is both a positive relationship in duration, which can be seen as our blue line right here, and occurrence of grooming behaviors with the amount of gill scars present on a crayfish. Finally, just to make sure that there was no interactions or that temperature was not playing a major role or a bigger role than gill scarring, we ran another ANCOVA with our independent variables being temperature, gill scar, and a temperature gill scar interaction, with our dependents being occurrence and duration, and species being a covariate. And the results of this ANCOVA indicated that there was no interaction effect and that only gill scars were actually a significant variable in predicting the occurrence and duration of grooming behavior. So, to wrap all of this up, what does this mean and how does this relate to our different objectives? Now, if you remember correctly, we had originally predicted that an increase in body size would be correlated with an increase in density, and we actually did not find that, despite previous literature showing that body size was related with the amount of symbionts. We did not find that at all, which suggests that maybe there are potential differences in grooming efficiency that are correlating with body size. So maybe the adults are much more efficient than the juveniles at actually getting rid of these symbionts when they actually do groom. For our second objective, we looked at if gill scarring was related to symbiont density, and that prediction was actually correct. So we found that symbionts may be, based on these results, symbionts may be the cause of this gill scarring, or as we haven't directly tested this, gill scarring may actually reduce the immune function and make it easier for those symbionts to actually latch on and establish on their host. For our next objective, we looked at the impacts of temperature on grooming behavior, where we predicted that increased temperature would actually increase the frequency and duration of grooming behavior. And what we found was that there was an increase or a decrease in grooming, but there was actually an increase in locomotion, suggesting a trade-off. So they seem to be grooming much less as temperature increases, and but they are moving much more. So it seems to be that there are some behavioral trade-offs based on temperature that we need to look further into. We then looked at symbiont density's relationship to grooming frequency and duration, where we predicted that symbiont density would increase, so would grooming frequency and duration. And although we did see some sort of relationship, is not what we expected. We saw a relatively negative relationship, even though it was not significant. And this may be explained by the fact that symbiont density is relatively only a snapshot of the actual symbiont's effect at that given time, and may not accurately demonstrate the impact that they have on grooming. So to wrap back around to that, we looked at gill scarring, which is more of a collective impact of symbionts on crayfish, and we predicted that increased gill scarring would actually increase grooming frequency and duration, and we did actually find this to be true. So while gill scars represent long-term symbiont effects, we may be seeing this due to the fact that gill damage may be impacting gill function and may actually become more important to groom when you have less functional gills present. Uh, so it may be more important to actually remove symbionts so you reduce the accumulation of that damage to your gills. Now, where does this leave us? What future directions do we want to go in? So, based on our results, we've actually expanded the study over a much larger temperature range, ranging from 55 to 95 degrees Fahrenheit, and with a much larger sample size. This should allow us to actually look at temperature's impacts much better than the short range that we did before. But we also wanted to expand into a couple different things. So we saw increases in locomotion and agitation in those crayfish. So we want to look at how temperature actually impacts agnostic behaviors, or agonistic behaviors, I'm sorry. Uh, so that's one of the studies that we are looking into, as well as looking at the thermal physiology, doing some population surveys, and mathematical modeling. So what my group is now working on is a number of different studies. The first one being our behavior study that I've just talked about, where we beefed up our temperature ranges and sample size to actually look more at grooming and how temperature impacts it, as well as at agonistic behaviors, and where we're going to be looking at the effects of both temperature, which is related to climate change, and density, which actually is a proxy for habitat loss. On top of that, we've started doing a number of physiological studies, primarily looking at the critical thermal limits of both of our species of crayfish, as well as brachiopodellids. And preliminary results are actually showing what we expected, being that Cambarus bartoni has a relatively larger thermal breadth than that of Cambarus latymanus. And we're also going to be looking at oxygen metabolism, specifically looking at how temperature impacts that, but also how symbionts impact that by testing crayfish with and without their symbionts to see if there's any impact on their physiology. On top of that, we have a team that is currently working on our field ecology studies. So we are doing a number of different population surveys across six different streams within the Clemson experimental forests, where we're doing a mark recapture studies, which you can actually see on the left. We have a crayfish right here we tagged with our fluorescent tag, which will allow us to actually look at the seasonal impacts on population of crayfish and brachiopodellids, and also just look at the population health of all of these species together. Using all of these, then, we are planning on doing a mathematical model where we're able to actually predict the impacts of climate change and temperature warming on symbiotic relationship outcomes. Now, a quick plug, because I do want to say this, and this is part of my team's work, but my climate change symbiosis CI is actually recruiting for next year, where we have a number of different teams. So if any of these studies I have talked about are of interest to you, please reach out. We have our field ecology team, behavioral ecology team. We have a team focusing on dissections, as well as a team working on physiology. And with that, I would like to acknowledge all of my funding sources from Sigma Xi, the American Museum of Natural History, Clemson Graduate Student Government, SICB, as well as Creative Inquiry and the Department of Biological Sciences, as well as all of these individuals and institutions. And with that, I will take any questions.